was funded through a NOAA Ruth Gates Restoration Innovation Grant. Okay, just to give you a little bit of a background, I'm based on the island of Saipan, which is part of the Commonwealth of the Northern Mariana Islands. The CNMI is located in the western North Pacific Ocean, just north of Guam and to the west of the Mariana Trench. And unfortunately, the CNMI, the corals of the CNMI and the people of the CNMI had a pretty rough 5 years from 2013 to 2018. We had 3 severe thermal stress and mass bleaching events and 2 Category 4 and 5 super typhoons that hit our islands over that 5-year period. And as a result of these disturbances, we lost over 60% coral cover on our shallow 4 reef sites and lagoon sites on Saipan, which resulted in huge changes in coral community structure and losses in ecosystem services. And so following this rapid decline in our coral reef ecosystem, local managers and stakeholders really started focusing their attention on reef restoration to try to mitigate some of these losses of really valuable ecosystem services. So as most of you here know, there are many approaches to coral reef restoration. Most of them involve some sort of asexual or sexual propagation of corals. We are going to focus here on sexual propagation. So sexual propagation, by harnessing the vast amount of reproductive output of broadcast spawning corals, sexual propagation has the potential to really greatly increase the scale of restoration and also the genetic diversity and subsequent adaptive capacity of restored coral populations, which is really important in the face of ongoing climate change. So sexual propagation, again, has these benefits, but it has some challenges as well. Gamete collection and larval culture can be logistically challenging, especially at larger scales, and outplanting and survival is still a bottleneck in the process. And so many organizations, including our collaborators, Seacorps International, have been trying to solve these problems for over a decade now, and they've made huge advances in the field. And one of the tools that they've developed, as you just saw, a great introduction of, are these settlement substrates. This is their original concrete tetrapod design, but there's been other iterations of these substrates. And so the idea is that larvae settle on them, and then divers can quickly outplant them on the reef without any need for a fixative, like epoxy or any other fixative. So they're rapidly outplanted and they self-stabilize in the reef structure. And so this can be really effective, as you just saw, but these substrates are fairly large. They're about the size of your fist. And if you think about really upscaling this process, handling, transporting, and dispersing these larger substrates could get a little unwieldy. So we started to think that maybe to go really large scale, we needed to think smaller, and I'm glad someone recognized that reference. I showed this slide to a bunch of high school kids, and they had no idea what that was about. But so the idea is, like many reefs, especially degraded reefs, have a lot of little holes, like smaller holes and nooks and crannies in the reef, where substrates could potentially stabilize in on their own. And also, on these larger substrates, the goal really is to have one surviving recruit that grows into an adult colony. And so maybe we can trim away a little bit of this excess substrate and develop some smaller substrates. And so that's where the idea of the larval seed beads came along. These are the OG LSBs. I made these out of epoxy and painted them so you could see them in photographs a little better. But the idea here was just to sort of figure out the size, a good size for these little nooks and crannies in the reef, and whether they would actually stabilize in these holes. And so, the overall idea being that they would be smaller, easier to handle at large quantities and distribute over large spatial scales with little to even potentially no diver input if we start thinking about autonomous vehicles dispersing these substrates. And so luckily we got some funding from the Ruth Gates Coral Restoration Innovation Grants that allowed us to really develop this idea and produce actual real substrates. And so they started with some really bad sketches. Thanks to Miles at C-Core, he somehow made these into really cool 3D models and engineering designs. And then ultimately we ended up producing a subset of 3 of these designs for testing. And so we started out, and we had to call them something. So we have the Death Star, which is a round substrate that's smooth on the outside with square holes. The mini gear that looks like a little gear, and the gear pod, which is, has the ridges but kind of looks like a little seed pod of some sort. So these are about 1 to 1.5 centimeters maximum length. They're made of 95% alumina ceramic. And then so once we developed them, we had them in our hands, and we needed to move forward with initial testing of settlement rates, survival, and reef retention. And so we've just started this process in the last couple years. So our initial testing happened in May of 2021. We collected gametes from Acropora pulchra colonies in the Saipan Lagoon. We assisted fertilization and ended up putting about 2,000 embryos in 12 replicate chrysal bins in an ex situ seawater system. So after about 4 days, these larvae were swimming around the bottom and were competent to settle. So we placed 15 each of our LSB designs and 3 of the larger C-Core, ceramic C-Core stars in each of these bins for a choice experiment. We allowed them 4 days to settle. After 4 days, we removed any larvae that were left and counted all of the settlers using a microscope and fluorescence adapter. And so using these counts, we calculated Manley's selectivity index to assess substrate preference based on the proportion of available surface area and the proportion of settled larvae for each substrate. And this pink line essentially represents the values we would expect to see if they were settling indiscriminately on these substrates based on their availability in the environment. So if the 95% confidence intervals overlap with that line, they're just settling on them based on their availability. If it's above that line, they're actively choosing, they're selecting those substrates. And if it's below that line, then they're relatively avoiding those substrates. So we found that there was, the Acropora pulchra had a strong preference for the gear pod design. And they were settling on the Death Stars sort of relative to their abundance, and they were relatively actively avoiding the mini gear design and the C-Core star. So there's a lot going on here, but if we look at the top right graph, we have the proportion of beads, LSBs, with at least one settler, which is the pink bars, and those without any settlers, which is the teal portion of the bars. And the bottom left, we have our mean settler density, so the number of settlers per LSB. And we can see through both of these, the gear pod is performing better than the other 2 designs. We have almost all of them had at least one settler, and the density of settlers was much higher on the gear pod. So we had almost 23 settlers per bead of the gear pod compared to about 10 or 11 on the other 2 designs. And this resulted in much larger total settled juveniles produced on the gear pods than the other 2 designs. We then packaged these LSBs up in these stainless steel mesh little packets. If anyone has better ideas how to contain these things, let me know. And then we brought them out to our ocean-based nursery for 5 months for a grow-out period. After 5 months, we brought them back into the lab and we counted all of the surviving juvenile corals. And so again, these are the same graphs we have with survival. We can see that the gear pods are again outperforming the other 2 substrate designs. They had many, if we look at, we estimated survival based on the mean number of settlers per bead when we put them out and then when we collected them again. So the Death Stars had much lower survival at about 5%. The gear pods had the highest survival at about 18%, and also much higher juvenile density per bead. So we had four settlers per bead still after 5 months, whereas the other 2 survival was lower. And again, this resulted in higher overall production of juveniles on the gear pod design compared to the other two. So we tested, and then in July of the same year, we did some preliminary tests with 3 other species, Acropora circulosa, Acropora tenuis, and Goniaster retiformis. And this is, again, the selectivity index. We're missing the mini gear here due to a logistical fail on my part. So we only have the 2 bead designs and the Secor stars. But interestingly, the 2 acroporid species had very similar preferences to each other, and similar also to the Acropora pulchra that we had tested previously. They also really preferred the gear pod over the other 2 substrate designs. The Goniaster retiformis, on the other hand, there's a lot more variability, but they were not as they were indiscriminate in their settlement. They were just settling on the substrates based on the availability in the bins. And so we've done some preliminary outplanting, some pilot outplanting. I don't know if you can even see them. The arrows in the top right show some beads in the reef substrate. They really just fit in these holes and get incorporated into the reef. And then we have some babies growing here. Some of them have started to overgrow onto the substrate, which was really exciting. But we're still following these guys in the fields. So in conclusion, our gear pod in these trials, our gear pod design was the most effective in terms of yield and production of juvenile corals. Seemingly small differences in microhabitat characteristics resulted in large differences in overall yield and production. Settlement preferences seem to be taxa specific, so that's something I should keep in mind in our restoration practices. And while we realize that these beads might not work on every reef in every environment, these preliminary results are pretty promising that they could become a really valuable tool in large-scale coral reef restoration. So we're going to continue with our research and development on our current and new designs. We really want to reduce the cost a bit. They're currently at about 20 cents each. We want to get that down to about 10 cents each. We're going to continue monitoring our reef retention and survival experiments over various environmental conditions, test different species in an upscale outplanting, and I really want to thank all of the team, the volunteers, staff, and interns that really made our 2021 spawning season a success. Thank you.
Thank you very much, Lyza. Speaker's doing a good job of staying on time. So we have time for a question or 2 from the floor. Yes, here.
It's excellent. I'm very excited about this.
Okay.
I wonder how much that gear pod, if it's neutral, or how much it weighs.
Oh, you know, I do know how much it weighs, but I can't think of it off.
Buoyant?
No, they're negatively buoyant. They sink.
Thank you so much.
Mm-hmm. Oh, sorry.
Quickly.
Did you observe like differences in growth post-settlement between each, substrate?
So unfortunately, at this point, we haven't tracked growth of the individual recruits, but that's something we hope to do in the future.
One more question, maybe while we get the left.
Okay.
You're the only one with your hand up, so.
You know,